Hi viewers, welcome to our channel. Okay, still on genetics part 2. Just like we described some of the inheritance that deviated from Mendelian's principle. Another one is a mutation, but we need knowledge of chemical structure of DNA and RNA to understand the concept of mutation. Like what is mutation? Mutation is a sudden change in the genetic composition, sudden change in nitrogenous base. Those genetic composition are those bases. If they are not complementary paired, it will lead to mutation of those genes. Even though we have various types of mutation, but we'll just stick to mutation at this level, at this level biology. So we look at mutation as a sudden change in the nitrogenous base. Sudden change. Once there is a sudden change in the base sequence or nitrogenous base. So, but when we say we need concept of chemical structure of DNA and RNA to understand this concept of mutation. Now, in DNA analysis, DNA, what does DNA mean? What does the full meaning? We say DNA. What is the full meaning of DNA? Students should know the full meaning of DNA. That's the first aspect of biology. That's the beginning of biology should know the meaning of DNA. That is the oxyribonucleic acid, kind from DNA. That is RNA. That is DNA. The other aspect of a genetic material is RNA. That one, just like DNA, that one is ribonucleic acid. Unlike DNA, unlike RNA, DNA does not have oxygen at the ribose sugar. That's the name. Deoxyribonucleic is deoxygenated in the position of the ribose or pentose sugar. Okay, let's look at the chemical structure for it. We have basically chemical structure of DNA. First of all, the chemical structure of DNA is a nucleotide comprising of the following: comprising of a phosphate backbone, and then a pentose sugar. What is the pentose sugar? Sugar with 5 carbon. Example of sugar with 5 carbon is ribose. We got this. And then the last is in nitrogenous base. Two bases are known. One is purine base, and the other one is pyrimidine. Two purines are known. One is adenine, and the other one is guanine. Two pyrimidine bases are known. One is thymine, and the other one is cytosine for DNA. So in the chemical structure of DNA, these two actually forms a backbone. And these two are the main concept of DNA, that is the nitrogenous bases, which are purines and pyrimidine. In fact, in DNA, purines must pair with pyrimidine via a bond called hydrogen bond in the bases. Okay, now but the purines pair must be a complementary pair, as in one base must complement the other. Failure on that issue of complementation will result to gene mutation. Like now, it is a must that a purine base like adenine, a purine base like adenine must pair, adenine A must pair via a hydrogen bond with thymine T. So that means all T must complementary pair with A. So this is purine base, this is pyrimidine. Or on the other hand, a guanine purine base must pair with a pyrimidine cytosine. That is G will pair to C, or C will pair to G. Okay, you will see a question. Most of your exam questions, WAEC, JAM, NICO, which of the following base sequence is complementary to TCA? Which will be complementary to this base sequence? Which will be complementary, as in what base will be complementary to this base? Thymine, cytosine, adenine. So anyway, thymine of a pyrimidine will only pair with adenine. So this will complement with this will complement to this, and this will complement to this. So this will be complementary to this. And this is what we call codon. Triplets. Triplets of nitrogenous base. This is complementary. So in the structure of DNA is a double helical structure like protein. It will also look like a mutated protein. This is the structure of DNA. And all this particular part of the DNA is the sugar backbone. Sugar backbone. So this is where you have ATG. Okay, if you have ATG, so ATG will always go with TGA, and again GAC. So if peradventure that this side is not complementary, maybe I have A instead of A to T, I have A to C instead of C to G. So this particular place is called mutation, and most time we can call it gene mutation. So anything that is suddenly changed, if one base is not complementary to the other one, it will result to gene mutation. And most of these gene mutations are transmittable. They can pass on from parents to offspring. So this is what it is, gene mutation, from the structure of your DNA and RNA. DNA differs from RNA in that RNA, just like DNA, is double-stranded, RNA is just single-stranded. Just like DNA is deoxygenated in position 2 of its sugar ribose, RNA is oxygenated at that same position. A thin line is what is separating these two genetic material. A thin line. It's just thin line. The sugars are the same, cells are the same. Only that this one lacks oxygen, one has oxygen. Only that the one of the pyrimidine bases are substituted by another purine base. So in RNA, uracil replaces cytosine. So difference is the uracil. So what it simply means that any other base remains the same. So if this is to be RNA, so anyway I see cytosine, it will be changed with uracil. So now, what will be complementary? If I see a DNA strand, that we know this is a DNA strand, this is a DNA strand. This is an RNA strand because uracil is one of the purine base. You don't see any uracil in any strand of DNA. What replaces uracil is cytosine. What replaces cytosine in RNA is uracil. So this uracil will be complementary to this uracil will be complementary to G, it will be complementary to U, it will be complementary to what? To what? T. So it means that UGA complementary base sequence of RNA is complementary to GUT. So UGA is GUT, while TCA is AGT, while AGT and ACT or TAC is DNA, UGA and GUT are RNA. That's just like I said, this is what they separate these two genetic materials is just a thin line, just slight differences. One has this type of base, both of them are nucleotide. Nucleotide in the sense that both has the nitrogenous base, both has phosphate, and both have this thing. So they are all nucleotide and genetic material of most organisms. These are on the age of the mutation. Okay, at this point, I think this is where we come to an end in this genetic part 2. In part 3 of this topic, where we're going to round it off, we're going to be discussing application of genetics, and most especially in the area of medicine, in the area of agriculture, and in the area of biotechnology. Thank you once again, our viewers.